Hello, my name is Sam Turco. I'm a professor of biochemistry at the University of Kentucky Medical School. I've been at that institution for many years. As far as Kaplan Medical, this is my 17th year of doing reviews for Kaplan Medical. Chapter 8 deals with the high-yield topics of amino acid proteins and enzymes. There are 20 amino acids, and all amino acids have a carbon atom attached to a carboxyl group, an amino group, and a hydrogen atom attached to a particular carbon. Now this carbon is referred to as the alpha carbon, and the alpha carbon is always carbon 2. So in this figure, here is the alpha carbon shown in the yellow. Attached to the alpha carbon, shown here outlined in the white box, is the carboxyl group. So the carboxyl group is referred to as an alpha carboxyl group because it's attached to the alpha carbon. Also attached to the alpha carbon is an amino group outlined in the white box attached to the alpha carbon. So it's referred to as an alpha amino group. And also attached to the alpha carbon is a hydrogen atom. And also attached to the alpha carbon is an R group. So all 20 amino acids have an alpha carboxyl group, alpha amino group, a hydrogen atom attached to the alpha carbon, and what distinguishes the amino acids from one another is the R group, and the R group can either be a hydrophobic group, like aliphatic chain, or attached to the alpha carbon, the R group can be a hydrophilic group, carboxyl groups, amino groups, something along that line. So all 20 amino acids are distinguished by what else is attached to the alpha carbon regarding the R group. All 20 amino acids have alpha carboxyl group, alpha amino groups, hydrogen, as well as the R group, which distinguishes all 20 amino acids from one another. Shown here is a grouping of the hydrophobic amino acids. These are the amino acids in which the R group is a hydrophobic R group. And of these, there are two subclasses: aliphatic side chain amino acids, glycine, alanine, valine, leucine, isoleucine, and proline. And then the other subclass are the aromatic side chain amino acids, phenylalanine, tyrosine, and tryptophan. Now, in proteins, the domains of these amino acids that are hydrophobic are located in proteins that are inside the protein to protect the amino acids from the aqueous environment. Because hydrophobic side chains, they don't like water, and so they stay on the inside of proteins. Let's say globular proteins will have hydrophobic amino acids on the interior. Where else do you see hydrophobic amino acids in proteins? Those proteins that are embedded in membranes. Part of the protein that spans the membrane is composed of hydrophobic amino acids. That can be any of these shown above. And lastly, where do you see hydrophobic amino acids? These are amino acids that are in proteins that will be destined to be secreted, membrane bound, or lysosomal. That was discussed in Chapter 4 in the Biochemistry lecture notes. Remember that proteins that are going to be secreted, membrane bound, or lysosomal must have an N-terminal hydrophobic signal sequence. This is a sequence of about 10 to 15 hydrophobic amino acids. And why they must be hydrophobic amino acids? Because the signal sequence is what's being recognized by the signal recognition particle in the cytoplasm. The SRP, signal recognition particle, sees the stretch of hydrophobic amino acids and attaches to the hydrophobic peptide region, and then directs that protein to the outer leaflet of the endoplasmic reticulum, and then a pore is opened up, and then that stretch of hydrophobic amino acids is directed into the lumen of the ER. So that must be an N-terminal hydrophobic signal sequence composed of these amino acids shown here at the top of the figure. In this slide are the hydrophobic amino acids: glycine, alanine, valine, leucine, isoleucine, and proline. Now all 6 of these amino acids, including all 20 amino acids, have that alpha carboxyl group, to which an alpha carboxyl group is attached to the alpha carbon, and the alpha amino group also is attached to the alpha carbon. And where these amino acids differ is the side chain, the R group. And the smallest of these R groups is a hydrogen, and that amino acid is glycine. So what I'd like to do in this slide is just highlight one item that you might see on a board exams if you see that particular amino acid in some sort of vignette. What do you automatically think of if they talked about glycine, for example, on the board exam? So glycine has the smallest R group, and what you automatically think of regarding glycine is collagen. Glycine dominates collagen. Every third amino acid in collagen is glycine, but it's not a hallmark of collagen because glycine virtually is in all proteins, but it dominates collagen. So if you see glycine on board exams, think collagen. The next amino acid to talk about is alanine. Alanine has a methyl group attached to that alpha carbon, highlighted in the yellow. And alanine, if you see alanine on board exams, you automatically think that they might be asking me about starvation conditions. And the reason is because alanine is a glucogenic amino acid, and when muscle protein gets broken down under starvation conditions, the glucogenic amino acids will funnel their way down to alanine. Alanine will leave the muscle cells, get into the bloodstream, and from there, the alanine will head over to the liver, and in the liver, the amino group is removed to make urea, and the 3-carbon skeleton is pyruvate, which then goes into the pathway of gluconeogenesis to try to maintain blood glucose. The next 3 amino acids are valine, leucine, and isoleucine. Now, don't go memorizing the structural differences between valine, leucine, and isoleucine. What's important to note is that the side chain is an aliphatic branched chain. And these 3 amino acids, if you see them together on board exams, you automatically think the major disease called maple syrup urine disease. So all 3 of these amino acids will be affected in that disease. And then the last amino acid to review is proline. See, the R group of proline is a ring, and there is an enzyme called proline hydroxylase that uses vitamin C to put a hydroxy group onto proline and to make hydroxyproline as a product. Now, if you see hydroxyproline on the board exams, you automatically think collagen. And it should be pointed out that hydroxyproline is found only in collagen. It is a hallmark of collagen because the only protein that has hydroxyproline is collagen. This next slide are the hydrophobic amino acids phenylalanine, tyrosine, and tryptophan that have aromatic side chains. So of these 3 amino acids, the 2 on the left, phenylalanine and tyrosine, don't go memorizing structures, but notice the difference between the 2 is the hydroxy group. Again, these 3 amino acids have alpha carbox group, alpha amino groups attached to the alpha carbon. Let me show you the alpha carbon once again. Here outlined in the yellow circle is the alpha carbon. Attached to it, the carbox group, amino group, the hydrogen, and the side chain. And the side chain is an aromatic side chain. So again, the lower left 2 amino acids, phenylalanine and tyrosine, the difference is a hydroxy group. So on board exams, what they might ask you about is a major enzyme that's involved in converting phenylalanine to tyrosine. If you know there's a hydroxy group on tyrosine and phenylalanine does not have it, the name of the enzyme clearly must be phenylalanine hydroxylase that puts the hydroxy group on phenylalanine to form tyrosine. And when that enzyme is defective, it gives rise to the major disease called phenylketonuria, PKU. Another reason why you might see phenylalanine and tyrosine together on national board exams is because both amino acids are precursors of catecholamines, epinephrine, norepinephrine, and so on. The next amino acid on the right side is tryptophan. If you see tryptophan on a national board exam, what they might ask you about in that question, it might have something to do with serotonin synthesis or niacin synthesis, and you should know that tryptophan is used to make serotonin as well as niacin. It's also used for other reasons to make melatonin, but on board exams that's unlikely to be. Most likely it would be serotonin and niacin if they're asking you something specifically about tryptophan. The next grouping of amino acids we're going to be talking about and showing you some structure are the hydrophilic amino acids. And of the hydrophilic amino acids, there are three subclasses. One is the amino acids that have positive charged side chains, and there are three of them: lysine, arginine, and histidine. There are two amino acids that have negatively charged R groups: aspartic acid and glutamate. And there are six amino acids that have polar uncharged R groups: serine, threonine, cysteine, methionine, asparagine, and glutamine. Now, where do you find these hydrophilic amino acids in proteins? In contrast to the hydrophobic amino acids, which are found in the inside of globular proteins, the hydrophilic amino acids are found on the outside. They're found on the periphery of proteins because that's where water is encountered, on the periphery, the external side of proteins. They face the aqueous environment. And also some of the hydrophilic amino acids are found on the surface of proteins to undergo modification. Modifications such as protein phosphorylation that might occur on serine and threonine that we'll be talking about shortly. The first grouping of the hydrophilic amino acids to talk about are the ones that are positively charged: lysine, arginine, and histidine. And one thing I want to stress about the positively charged amino acids, you should know that lysine and arginine are strongly positively charged and histidine is weakly positively charged. Now, I know in some textbooks they spend a lot of time talking about pKa's and isoelectric point and so on. For board exams, those are low-yield items. Don't waste your time understanding issues of pKa's and PI's because they are unlikely, I cannot guarantee it, but unlikely to be on national board. Don't waste your time understanding pKa's and so on. Now, another item about the positively charged amino acids, don't go worrying about the exact structure. What's the structural difference between lysine and arginine? Just know they are strongly positively charged. So if you're going to see something about lysine and arginine on a national board exam, one thing you might think about is that these are amino acids that you would find that dominate histones. Histones are strongly positively charged proteins that are found in the nucleus. And one characteristic, they are dominated by lysine and arginine, not histidine. You don't find much histidine in histones, even though the names, the words sound very similar. Histidine is not found in appreciable amounts in histones because it is weakly positively charged, whereas lysine and arginine are strongly positively charged. Here is the alpha carbon once again. And when it's attached to the alpha carbon is the alpha carbox group, alpha amino group, and the proton and the side chain. Now, lysine, if you see lysine on a national board exam all by itself, independent of arginine, then you don't think histones. If you see lysine on a national board, what you automatically think of, or you should think of, and that is the amino acid hydroxylysine. So just like hydroxyproline that we just talked about, which is hydroxylated by the enzyme proline hydroxylase that requires vitamin C, lysine hydroxylase in fibroblasts use vitamin C to hydroxylate lysine residues, forming hydroxylysine. You must know that. That is a very high yield item for the national board exam. Lysine oxidase hydroxylates lysine using vitamin C. The next amino acid to talk about is arginine. And if you see arginine on board exams, without knowing the exact structure of arginine, what might you think of? And the answer is that arginine is used to make urea. Again, the structure of urea will be referred to later on in urea cycle in chapter 17. But in this chapter, just know that urea comes from arginine. Look at the R group of arginine with those nitrogens in the R group. That will end up being urea in the urea cycle. Now, there are other reasons why you might see arginine on national board exams. Besides being found in proteins used to make urea, you might see it for other reasons. You might see arginine, for example, as a precursor of nitric oxide, causing vasodilation. But here, you must know used to make urea. Histidine. If you see histidine in a vignette on a national board exam, what might you think of? You think of the possibility that histidine is in the active site of all enzymes. Histidine is involved in catalysis, carrying out the conversion of a substrate to a product. Now, another reason why you might see histidine on board exams is it's used to make the biological active molecule called histamine. The next group of amino acids that are hydrophilic are the two amino acids that are negatively charged, aspartic acid and glutamic acid. And at the lower left side is aspartate, on the right side is glutamate. Let me emphasize, by the way, the suffix, the ate ending. The ate ending is referring to the salt form of the acid. So aspartic acid is the same thing as aspartate. It just depends what the pH is. Glutamic acid is the same thing as glutamate. The ate is the salt form of glutamic acid, but they are one and the same molecule, but it depends on the pH whether it's ionized or not. Aspartic acid and glutamic acid. Here comes that alpha carbon once again. Let me show it outlined in the yellow. That is the alpha carbon. Attached to the alpha carbon is the alpha carbox group, alpha amino group, as well as the proton, and the R group has a carbox group. And you should know for board exams to some extent that aspartic acid has 4 carbons, and glutamate is 1 carbon larger. It's 5 carbons in length. But on the periphery of the R group is the carbox group, which makes these two amino acids negatively charged. So if on board exams you see aspartic acid in the vignette, what do you think they might be asking you about? What do you think the topic might be? And the answer is likely they're asking you something again about the urea cycle. Because if you notice the amino group in aspartic acid, that amino group will end up being one of the 2 nitrogens in urea. So if you see aspartic acid on boards, they might be asking you something about the urea cycle. On the right side, glutamate. Glutamate is needed for many, many things. For example, glutamate is involved in vision, but most likely if they're asking you something about glutamate on board exams, they are asking you about GABA, gamma amino butyric acid. Because in the conversion of glutamate to GABA, there is an enzyme glutamate decarboxylase, and what the enzyme does, it removes the carbox group that's attached to the alpha carbon, and by removing that carbox group, the product is gamma amino butyric acid as the product. But glutamate is needed for many reasons: vision, synthesis of GABA, and so on. But you must know glutamate for board exams because it's needed for many things. The last group of amino acids that are hydrophilic are those that have polar uncharged R groups. And there are 6 of these: serine, threonine, cysteine, methionine, asparagine, and glutamine at the bottom. Of these 6 amino acids, here comes the alpha carbon, once again highlighted in the yellow circle. There is the alpha carbon. Attached to it, once again, I keep emphasizing, is a carbox group, amino group, and a proton. And what distinguishes these amino acids is the R group, which is polar uncharged. Now, of these 6, 2 of them, serine and threonine, are noted for having hydrox groups. Don't go memorizing structural differences, but you should know that serine and threonine have hydrox groups in the R groups. And if you see serine and threonine on board exams, what might you think of? You think that these are sites, the hydrox group is sites for a process called O-glycosylation that was discussed earlier on in chapter 4 in molecular biology. When sugar chains, glycosylation, sugar chains are added onto proteins in O-glycosylation. Those sugar chains go on to the hydrox groups of either serine or threonine. And if you were asked what's an example of a protein in the human body that undergoes O-glycosylation, there are many proteins that undergo O-glycosylation. For example, collagen. For example, proteoglycans, chondroitin sulfate, hyaluronic acid, heparin undergo O-glycosylation. Mucins undergo O-glycosylation. There are many proteins in the human body that have sugar chains attached to them, and if they're attached to the hydroxy group of serine or threonine, that's referred to as O-glycosylation because the O is referring to the hydroxy group. The next two amino acids to review are cysteine and methionine, and they are noted because they have sulfurs in them. And you should know that cysteine, its sulfur is a sulfhydryl group. In methionine, its sulfur is not a sulfhydryl group. There is a methyl group attached to the sulfur. If you see cysteine on board exams, what do you think the topic might be? What do you think they might be referring to on the board exams? And you answer by saying that cysteine is involved in forming disulfide dimers. Two cysteines can react with each other, and the sulfurs of the two cysteines form a disulfide dimer, and disulfide dimers are helping to maintain tertiary structure of many, many proteins. Methionine does not have that sulfhydryl group, so it's not involved in formation of disulfide dimers. And what might you see methionine involved in on a vignette? And the answer is it's used to make S-adenosylmethionine. And what's S-adenosylmethionine? It is a methyl donor. Notice the methyl group on the sulfur. When methionine is put onto adenosine, the product is called S-adenosylmethionine, and that methyl group becomes a methyl donor. To make what? To make things like epinephrine, to make lecithin, to make the 7-methyl G-cap on messenger RNA. Where does the methyl group come from? You're looking at it. It's on methionine, which is also part of the molecule called S-adenosylmethionine. The last two amino acids to review are asparagine and glutamine. Now, asparagine is, from the previous figure, if we take aspartate and put an amino group on the carbox group of the R group, it becomes asparagine. If we take an amino group and put it onto the carbox group of glutamate, which is 5 carbons, we form glutamine. So one is asparagine, one is glutamine. The difference is 1 carbon difference. Asparagine, if you see asparagine on board exams, what do you automatically think of? You should be thinking N-glycosylation. Just like O-glycosylation means there's an attachment of sugars onto the hydroxy group of proteins that have serine or threonine, asparagine in N-glycosylation, there are sugars attached to the nitrogen. See the nitrogen? The nitrogen is where sugar chains are added onto asparagines of specific proteins. If the boards were to ask you, what's an example of a protein that undergoes N-glycosylation, and you answer by saying virtually all proteins, not all of them, but virtually all proteins in the bloodstream, like all the blood clotting factors, all the immunoglobulins, all the receptors on cells, a lot of transporters, GLUT1, 2, 3, and 4, for example, the sodium-potassium pumps, they all have N-glycosylation, a major process that goes on in the body. And again, what that means is there are sugars attached to the nitrogen of asparagine of proteins. That's called N-glycosylation. Now, the last amino acid to review is glutamine. And glutamine, if there's some vignette on boards that talk about glutamine, what do you think of? You say to yourself, well, they're probably asking me something about nitrogen metabolism, because glutamine, with its nitrogen on the carboxyl group of glutamate to form glutamine, that when it's dumped into the bloodstream is it's transporting the nitrogen or ammonia, one and the same thing. It's bringing the ammonia to the kidney. So in the kidney, the amino group is removed, and the amino group then will be excreted into the urine. But where did the amino group originate from? It comes from glutamine. So when you think glutamine, you say to yourself, it is the major transport form of ammonia in the bloodstream, bringing the ammonia to the kidney for excretion. In the hemoglobinopathies, there are a number of problems that can go wrong with the hemoglobin. The most common one is hemoglobin S, in which at position 6 there is an A, the nucleotide A is converted to a T, the nucleotide T. And as a result, the codon for glutamate shifts for the codon for valine. And if we were to take the hemoglobin and electrophoresis hemoglobin S compared to hemoglobin A, because it has glutamate, would migrate to the positive terminus faster than hemoglobin S, which migrates slightly slower. So on the diagram on the right, we see hemoglobin A migrating to the positive terminus a little bit faster than hemoglobin S. And the reason for that is because the glutamate, shown in the yellow, is in the beta globin, whereas in hemoglobin S, there in the blue, there is valine. All the other amino acids are normal. Are the same in hemoglobin S as well as hemoglobin A. The only difference is glutamate. The valine, glutamate is negatively charged, as we just reviewed, whereas valine is hydrophobic amino acid, and because of the extra negative charge of the hemoglobin, migrates slightly faster than hemoglobin S. Now there's another variant of the hemoglobinopathies, and that's called hemoglobin C. Now in hemoglobin C, there is a lysine instead of glutamate. Now lysine, because you know structures of amino acids, lysine has an extra positive charge compared to glutamate. Then lysine, now the hemoglobin C will have slightly more positive characteristics than hemoglobin A, because hemoglobin A has glutamate, hemoglobin C has lysine. Lysine's positively charged, glutamate is negatively charged. So if you were to compare in electrophoresis, hemoglobin A versus hemoglobin C would migrate slower, migrating to the positive terminus compared to hemoglobin A, which has that negative charge, attracted to the positive terminus. Therefore, hemoglobin A migrates slightly faster than hemoglobin C. So if we were to take a patient who has hemoglobinopathy, and we wanted to know, was it because of that patient have hemoglobin S or hemoglobin C? All we got to do is take that person's hemoglobin and subject that protein to electrophoresis, and we would notice that in this particular example, that this patient has hemoglobin C, hemoglobinopathy, because that hemoglobin C is migrating side by side to hemoglobin C in the standards on the left side. So the left side are the standards, the right side is the patient, and the hemoglobin of the patient is migrating side by side to hemoglobin C. That patient therefore has hemoglobin C, meaning the mutation is resulting in a lysine for the glutamate.